everybody, for this afternoon session. We're going to be working around the clock to bring you the best of AI and biology. So gather around if you want to hear all about the impact that AI is having on the field of biology, on the field of synthetic biology. My name is John Cumbers, and I'm the CEO and the founder of SynBioBeta, and I'm also a board member of iGEM. So it's wonderful to be here, to be back at iGEM. Up on my phone, it came a 20-year photo from the first time that I participated in iGEM. So it's great to be back, and it's great to see so much energy in the room, and also so much energy for our panel today, all about AI and bio. So I want you all to join me in giving a huge round of applause for our panelists today, Sana, David, and Jonathan. Please give them all a big round of applause. So welcome. Let's just go down the line and give quick introductions. Sana, let's start with you, please.
Yeah, it's fantastic to be here. I'm Sana Zakaria. I'm a senior research leader at RAND Europe, heading up a team on emerging technologies. So it's a not-for-profit research organization, and the team's portfolio spans research on engineering biology, AI, and quantum technology, so very small portfolio, obviously. And I guess to kind of bring it to life, one of my recent flagship projects has been on codifying the functional and biological capabilities of AI-enabled biological design tools and assessing their misuse capabilities as well. Yeah, it's fantastic to be here. Excellent,
thank you. David?
Hi, everybody. I'm David. I'm from NVIDIA. I hope you know the company. So I'm leading strategic relationships for NVIDIA in healthcare life science. So anybody doing AI for drug discovery, new materials, medical device, you name it, new medicine, I would be very interested to talk to you and contribute here. Background is in bioinformatics, PhD, postdocs, and 10 years in pharma.
Wonderful. And last but not least, Jonathan.
Thank you very much. I'm Jonathan. I'm the co-founder and CEO of DeepLife. So DeepLife is a tech bio company based here in France, but having offices also around mainly Europe and US. And we are developing virtual model of cells, like a digital twin of cells that we leverage mainly to accelerate drug discovery by trying to find the best indication for the drugs that are today already existing. So it's a very interesting field, a lot of across actually AI and biology, where we try to implement the best models to challenge, to answer the challenge around biology today and drug discovery mainly. And my background is actually not in bio, neither in AI. It's in engineering. I was working in the aerospace and defense industry for a couple of years, and now we try to bring those approach back into the field of drug discovery to make those development more reliable.
Wonderful, excellent. Well, I'd like to start the panel with what's hot in AI and bio. And David, I'd like to start with you because NVIDIA has the BioNeMo platform. And as far as I understand, your goal is to integrate all of these models so that whenever people are running models, they're running them on NVIDIA compute, ultimately on NVIDIA chips. So you have quite a broad view of the whole model ecosystem. So I'm wondering, from your perspective, what's hot right now? What models are people using, and where are you seeing the most exciting things at the intersection of AI and biology?
Yeah. So what is hot is at the moment in the business area, and I'm very impressed by the number of companies I see here today, or let's say very early stage concept companies sometime, which are using some of the model we propose. So NVIDIA is not developing all the model of the world, but we make it easy to adopt, try, and basically test those models. Some of the modalities, let's say the data modalities, which are the most interesting these days, are proteins models. So I saw a lot of application here of ESM3, BOLT, the BOLT type of model from MIT, and others to design new antibody, nanobodies, protein, binder, PROTAC, you name it, therapies. And that's very encouraging. So first of all, I'm impressed that young people in this crowd are already using such model and are very proficient in it. And it's a testimony that, in a way, we need to keep our guards up when you work in pharma, because the field is moving extremely fast. And the limit of those young talent is their imagination. It is not anymore basically the technological advantage or the compute needs.
Wonderful. So we heard about bolts, we heard about ESM3. Sana, what models are you most excited about, and what are you seeing the most traction?
I think I would add to what David said. We are seeing a lot of traction in genome language models and protein language models, protein binders, so AlphaProteo, Evo, Evescape, viral mutation prediction. So a lot of these tools tend to be quite popular. The other thing we're seeing is a lot of integration between the narrow tools and LLMs, and also agentic systems and agentic capabilities. So we're seeing a lot of these things tethered together in frameworks like the BioNeMo framework, and there are multiple other workflows that are emerging, which is a really interesting, exciting, and sometimes concerning development for various reasons, but I think primarily it's quite an exciting endeavor.
Wonderful. And Jonathan, what about you? Are you using other people's models in biology, or are you developing your own, or what does the landscape look like for you, and what are you excited about?
Yeah, actually, I'm gonna jump on what Sana and Dave said. It's pretty exciting moment here in biology because we don't have to do everything ourselves, which is pretty great. And as mentioned, for example, not so long ago, you might find, for example, like an interesting drugs, but you have no clue if this drugs is answering an unmet need beside asking all the doctors around the planet what is going on. Today, you have also new tools that allow you to contextualize your discoveries, and this is very, very efficient. And so indeed, at DeepLife, we build our own models, but we also rely on models coming from other partners. And biology is very vast. So from modeling any like at the DNA level, transcriptomic level, protein level, or more, it's becoming very complex. So I think today it's an exciting moment where everyone is going into one same direction on developing each one his own model, and like I would say like concatenating all those approach makes stuff extremely exciting.
Wonderful. Well, I'd like to throw it out to the audience. I'd like a show of hands so we can see what kind of models people are using here. So put your hand up if you've used Evo or Evo 2. That's a smattering of folks. What about Evescape? What about bolts? What about ESM3? What about AlphaFold? Any others we should poll the audience on? What about BioNeMo? Who here has used BioNeMo?
Nice.
Any others we should poll the audience on?
What about the latest OpenFold? There was OpenFold 2, OpenFold 3 just got released.
Excellent. Anything else? Any models that we missed that people are finding particularly valuable right now? Shout them out. Say it again. RC? Odyssey. Anybody using Odyssey? Okay, well, I hope you wrote them all down. There'll be a test at the end, and we want to see hands-on practical experience. So that's very practical, what people are using now, and that was quite remarkable for those who didn't see. I mean, AlphaFold swept the floor there with how much utility it provides, or how many people have used it. Let's now move away from the present, what people are actively using AI for, and let's think about the future impact that AI is going to have on biology. I want you to think about 10 years into the future, and what kind of world do you think we're going to be living in? Sana, maybe you can take the lead.
So I get asked this quite a lot, and I've thought deeply about it. And I think there's going to be a real shift in how AI impacts biology. So last year, I think the conversations were quite grounded in things like AI and biology learning each other's language, where people were like, oh, we come from different systems, different cultures, it's really hard to integrate. And I think now we're seeing a bit of a co-evolution of these systems and tools and capabilities. So it feels a bit more integrated. So I think I think there are three things maybe that are going to shift quite significantly over the next decade. And one is going to be around the center of agency and the scientific endeavor. And that relationship between a scientist and AI as an assistive tool is going to change quite significantly, because we're no longer the only ones coming up with hypotheses. I think AI systems are also generating hypotheses, so the value of innovation will move beyond invention to also curating and selecting and supporting in direction setting the best ideas and candidate ideas. The other thing that I think we'll see in some of the talks that I went to today really drove that point home is we're probably going to see AI systems work across multiple modalities. So at the moment, we already have systems where AI can integrate, and in silico and wet lab capabilities are joined up. I think we're going to see that become more prevalent, almost using AI as a brute force optimization sort of machinery to kind of, test multiple ideas at a time. So we're going to see more of that integration. I think the third one might be we're going to see maybe some decentralization of biological capabilities. So maybe in 10 years, we might be able to have smaller systems, more autonomous systems producing therapeutics, biofabrication. We have a biofabrication arena here, but creating food materials, almost like 3D printers on steroids. So I think that's quite exciting. I think primarily we're going to see an expansion of the opportunity space and programmable biology potentially over the next 10 years.
Actually, that's the theme of SynBioBeta next year, is programmable biology. It's the Programmable Biology Summit. So
I didn't know that.
I'm glad you plugged that. It's going to be in May in San Jose. And we're also going to have a whole bunch of hands-on demos for people to bring their laptop and try out a lot of these different models. We have Thomas Kurzo, who was the author of the Boltz model from MIT. He's going to be coming to talk. You mentioned self-driving labs, so I'm curious which of the companies that are doing autonomous science or self-driving labs are you most excited about?
I mean,
or academic labs?
There aren't that many around yet, or they aren't very well known. I think the most well-known ones tend to be, people talk a lot about BenchLink, for example, as a capability to kind of plug your models into wet lab science, and that comes up quite a lot. But I think there's going to be more and more of those use cases emerge, which are quite niche, but actually it's really hard to find examples of that, to actually calibrate it against what's happening in the commercial space. So it's hard to answer that question. But yeah.
Yeah, I've seen a couple. Just recently, Google Life Sciences came out with an autonomous science paper and an interesting result. And then Future House, funded by Eric Schmidt, Sam Rodriguez spoke at SynBioBeta this year, talking all about that and the impact that that's going to have. And then there's a few interesting academic labs in the UK who've done some work on autonomous science. Doug Kell published a paper quite a number of years ago on that topic. So David, I'm curious, same question for you. Maybe you want to riff on anything that Sana said, but what does the 10-year horizon look like for NVIDIA?
For NVIDIA, I will try to not answer that and leave that to Jensen. But
okay.
I will say for the field of 10 years, ahead. It's a long time. You need to understand we got the Nobel Prize for AlphaFold last year. And that is quite impressive for an invention that just got popped out like a few years ago, 6 years ago. It's amazing. So the field is moving at an incredible speed, and the speed has increased since. And every year it's faster in terms of AI model. I think so when you talk and you take a step back about the field, I think we have the foundational building block, the foundational models for protein design, RNA design, genomic design, physical design of membrane, protein, etc. exist. What will be over the next 10 years is a consolidation of data. And there will be a data moat approach to build companies that will differentiate the company which will win and license basically access to that data. So I think It will be a combination of clearing out of the field. A bit of the noise will go and retreat a bit, and we'll see a bit of, okay, that's a winning approach. But I will also say in the next, I mean, it's not 10 years definitely, but the robotic lab in the loop, basically, is popping up very strong for us. We have, in the field of robotic lab, I mean, we have a tremendous amount of companies. So you mentioned a few, but there is a tremendous amount of company building robotic lab which are driven by AI experiments. And it's ready to buy right now. I mean, they come and they implement a robotic lab for you. You don't need even to buy the new material. It's just it fit within your office.
Drop some of the names. Who are you most
Automata
excited
Tech
about?
is a London-based company, Automata Tech.
Automatech.
Yeah, Automata Tech.
Automata.
You should not Google Automata. You never find them. You should tech at the end. Yeah, Automata Tech. That's a very good one. They are partnering with a lot of startups. There is another company, I mean, there is Medra in the US. And after that, you have the company which are building on such technology and combining their AI model with those automated labs and robotic labs. One of them being, for example, Lila Science in the Boston area. There is, even in France, there is Ictos is building basically some of those robotic lab approach. And it goes on, it goes on. Many companies are having a data generation strategy, and it involves robotics. You cannot scale up basically the lab work the good old way.
Wonderful.
And the technology exists.
And we'll come back to the data sets question because I'm really curious about that. The same question to you, Jonathan. What does your 10-year horizon look like, or what does the vision look like for DeepLife in 10 years' time?
Well, it's a very interesting question, very vague. There is actually part of what Dave and Sana were saying. There is actually interesting trends today on how the technology is going and where it's going, and we have also very strong constraint on how we can deploy this technology for actual use. So my answer would be more focused around drug discovery, but actually it could be way broader than that. Today at DeepLife, for example, we mainly work on model of cells. It's basically what we are doing. Everyone is focusing also on virtual cells, and like this is what we have to tackle first. But if you think about biology more in the context of drug discovery, way broader, well, a disease does not stop at the cell level. You have the tissue, you have the organ, you have everything that goes beyond. And now we start also having some data that help us try to navigate the complexity between the interaction between cells and cells, so cell-cell interaction, going way one step beyond and one step beyond all the time. So this is very interesting because we can come at a point where we have a completely explainable way of describing the effect of a disease on an organism. And if you go one step further, because this is just like kind of a generalist view, you say, okay, this is true for, let's say, human that I've seen into my data sets, but let's say now we find a way of accessing data that come from you, from your family, then you have the ability to build not a digital twin of a cell or an organism or a tissue. You can build like almost like a virtual representation of the disease within the right patient. And then you come to a personalized medicine where your model could be tuned with your own data. So it might not be like a 10-year vision. It might be maybe too ambitious for a 10-year vision, but it's basically where we go. And then the question that was remaining, and actually we are discussing that a little bit before this talk, is what remains to be done so those drugs can be approved. And this is, I think, the biggest challenge. How do you bring those drugs to clinics? Today we've been developing a lot of models. I think there is a lot of people here developing very interesting technologies that could be bring to the market. I mean, at least with a proof. Now this has to be approved as fast as it can be discovered. And this, I think, going to be a challenge. And hopefully within a 10-year time, we will have at least agreement on how we can fast track as well post-phase 1 drug development. That could be amazing.
Wonderful. Well, this wouldn't be an AI panel if we didn't ask the AI for its input on what it thinks is going to be the impact of biology and AI in the future. So at the end of the questions here, we're going to bring up 3 different models that we asked the question to, what is your prediction for what the future of AI and biology looks like? So stay around to the end of the panel and we will be getting input from the AI on that. Christina Agapakis, who used to be the creative director at Ginkgo, wrote a beautiful piece on her Substack recently saying that for many years we've been trying to use science to understand biology, and then came along synthetic biology and we were trying to use engineering to be able to create biology. And Feynman, who said, if I cannot create it, I do not understand it. So that's kind of been the mentality of synthetic biologists over the last decade, 2 decades. Now what we're seeing with the AI models is that, one, maybe we don't need to understand it from a scientific perspective. 2, maybe we don't need to build it from an engineering perspective, because now we can create these large language models, first of viral genomes and then of bacterial genomes and now of whole human genomes. And we can ask the AI, have a go at writing a whole genome for me, and then let's synthesize it and see whether it works or not. And we still don't need to understand it, and we haven't designed it. The AI maybe can't even explain why it chose the nucleotides that it did. So I think we're entering a brave new world of AI models for genome design. How soon before you think you can just type into a large language model the gene that you want or the genome that you want and get a pretty accurate result out? What do you think, David?
Pretty soon. I think my prediction would be that in a year we could generate a full human-sized genome. So the latest model, you mentioned it, Sana, is EVO2. EVO2 is limited. I mean, you can generate a fungi genome. I think you can. That's the max you can do with it. You don't go to bacterial even.
Which genome did you say?
Fungi.
A fungal genome.
A fungal genome, yeah. But definitely not a human. It's too large. There are limitations to that, the amount of data, but there is a hardware limitation also. So we don't have the material to do such thing. So it will evolve very quickly. The problem is we need a solution for that to invent the new medicine of the future, to design basically new therapies. Where would you inject your CRISPR medicine? Where would you design a new gene to compensate a particular trait for your disease? So it will happen. I think that will be in a year timeline. We'll see some evolution on that.
Sana?
I just want to say, so I think on EVO2, that's a great example because the recent publication, so EVO2 was used to create a bacteriophage, and it was selected and tested in the lab, and it was completely functional, and it performed better than sort of currently existing bacteriophages, which is fantastic for antimicrobial resistance. So I think we will get there pretty soon. I agree with that assessment. I guess I question, I think it's super exciting, but sometimes I deeply reflect on, should we be doing it just because we can? And how do we direct resources to things that are really useful and meaningful to solve big societal challenges and grand challenges and do more mission-oriented research? And I totally get the value of doing things for the sake itself, for the sake of discovery, but creating a whole genome, we sequence the whole genome, which is fantastic. We still haven't been able to create functional map of all of it. So we have the sequence, we have the data, we don't understand it. So.
And you work a lot with governments. So what are these the kinds of questions that governments are asking from the ethical point of view, from the societal point of view, about what capabilities we have and what we should be directing them towards?
I think a lot of the questions that I am in discussions with governments are around trade-offs for innovation versus risk, because biology is inherently dual use. So you want to drive innovation, you want to promote innovation, you want to see these capabilities and tools integrated into different sectors. At the same time, you want to understand what is the best place to apply some of these tools and capabilities, And what are some of the policy levers that we might want to put in place to manage any risks or unanticipated consequences from these? So those are the type of conversations that come about.
Yes, go ahead.
If I can contribute on, so it's not that we are not used to regulate technology. We do that since forever. We control when something is dangerous. So, and it's definitely true, we will have to regulate some use of those tools that we create. But I think the point I wanted to make is about the comprehension. So we might not understand what they are doing and how they do it completely, but I think it's a step forward to understand as humans, because we have limited capacity. We don't have the ability of maintaining the entire cell protein network and interaction, gene expression at the same time in our little brain. We need a tool to do that. We need a computer. So those tools will help us understand how does it work, how could it work, but we don't need to understand how the tool works completely. It's just we know to build it. But the tool inside, the statistical correlation is capturing, is maybe not so important, in fact. It's just a representation we can manipulate. That's what DeepLife is building.
Yeah, Jonathan.
Yeah, to add on top of that, indeed, I think I won't say it's a waste of time, but it's almost impossible now to try to understand how the model has been thinking. But we have methods to try to understand, to make the result explainable. And this is what is most important. So it's basically, regardless of how you find the result, if you can explain them, this is very important. And one of the focus indeed is, I think, developing those methods and make sure that we can always having like the confidence in the results that are going to be provided by such tools. So yeah, it's basically the point I have here.
So I want to move the conversation on to data sets, or back to data sets. We talked about the need for new data sets. We talked about them being a moat for companies that are raising money from investors. I'd like to know if each of you could wave a magic wand and produce a novel data set for biology that would move the whole field forward. What kind of data set are you looking for? What kind of experiments would you be doing? Maybe this is proprietary, Jonathan, but why don't you go first?
Well, recently, some data has been made available regarding like perturbation data. It's kind of a like sequencing of perturbation that has been CRISPRed or taste from drug on various cell lines.
Is this perturb-seq? Is that the technique? Yeah.
Yes.
So maybe just describe perturb-seq, because it's a pretty interesting technique for generating this kind of data.
So it's basically knocking out or some genes into particular cells and trying to see how the cell is kind of what is the response of the cell. So we sequence cells that has been CRISPRed or perturbed anyway. And the data that we collect out of it is very interesting. It's very, very interesting because it allows us to create perturbation in our models and see how they behave, and then it's a path forward for explainability in the models that we develop. But those data are kind of expensive. It's kind of a sequencing, so it's still expensive. You have a lot of, we have 20,000 genes. If you take on top of it all the drugs that we might want to use, it becomes like huge data sets that we have to do. One company did it recently. Like, I don't know if you're familiar with the Tau data set or something like this. One is kind of doing some experiments, but it's a huge amount of money. So if I have the magic wand, I would have done it for all the perturbation I have in my mind, which probably require an extra round of financing. But no, that's pretty cool. But another stuff that is important in the data is that maybe we will, maybe other panelists will discuss about it, is today we talk a lot about the volume of the data, but what we see today of interest is more like the heterogeneity of the data. So we need to have more diversity in the data that we have. For example, for us, it makes no sense to have like 100 times the same patient, one same cell type from a patient. We don't care so much. We prefer to have 100 patients. It's more interesting, but it's very hard to get those kind of very large-scale population embedded into the data representation. And if we go in that same direction of ethical issue on data representation, in drug discovery, most of the time, those data come from, let's say, European, American hospitals. So as long as you want to develop drugs for the whole planet, well, the representativity is not in your data. So I think it's that kind of stuff that if I had a magic wand, I would probably like to get those perturbation data on as much diverse human as I can.
Wonderful. Sana, what about you?
I've got a slightly left field answer. If I could wave a magic wand and create data sets, I would probably want to have neurodata and almost create big data for neurodata, because we're seeing a massive proliferation of brain-computer interfaces and their deployment in the commercial space as well. And brain data is highly variable, so it's almost the opposite problem where you have a lack of homogeneity in the data. So you need a huge amount of data and utilize AI to actually create some baselines. And I think that would be fantastic to be able to do.
And that's really interesting because this BCI data is plentiful if the BCI is working. There's one company, Paradromics, which is a competitor with Neuralink that's got some really interesting new data that's going to be coming out soon. So the BCI will, if it's working correct, will produce massive amounts of data, and it's really rich data as well, very different from genomic or sequence-based data.
Absolutely.
And then we saw NVIDIA as well recently raise, I think, more than $100 million for their mass spec data, which is also a very interesting new kind of data set that can be fed into AI models.
Yeah, I think the challenge in this space is that because there is so much variability and there is no baseline, we're already starting to use some of the neural data to think about cognition, to think about IQ, and really complex things that are really contentious, but we don't have a good data set to be really able to base any findings on or to make any assessments. And I think it's great for pathology, but it's generally great for understanding the concepts of cognition, for understanding, nature versus nurture, and all kinds of things in that space. And I think it's just going to be a new currency, neurodata, as well as genomic and wider omics data sets.
David, any thoughts from NVIDIA's perspective?
I think, to help the field going forward in data generation, we need to have a mechanism to represent the value of data set which are generated by companies on the balance sheet, on the P&L. At the moment, it's not represented. It's what you do with it, what you're able to sell, what product you're able to build and sell is represented on the P&L, but not as an asset. You cannot have a data set as an asset. It's also very hard these days to have a model as an asset on the P&L. And I think we should find a way to capture that somewhat. I think it's an asset like the others. It decays in value over time, but it's like, for example, clinical trial from 10 years ago are barely usable. It's not really usable at all. The standard of care has changed. The method, the machine have changed how to measure things. But patient population has changed. But also you don't treat people the same way anymore. So it's finished. You cannot use it. And that would encourage early investment if we can represent this value, because you need to create data to then basically develop models and then therapies, invent a new therapy. And that's, it's a catch-22. You cannot develop the model and go for a therapy if you don't have the data. And,
If we think about, we've covered models, we've covered data. What have we not covered? What are we missing that you think if we had it would move the whole field forward? Or what discussions do you think we're not having? Sana?
So I think more, not benchmarks in the way that benchmarks currently exist for assessing AI model capabilities, but almost more what you are doing, Jonathan, with creating digital twins. And I think we need more scalable digital twins because biology is super complex and it's really hard to simulate biology. And we are creating AI models that are have generative capabilities, predictive capabilities, but Again, a lot of these don't translate through to the wet lab quite often, or they don't translate through to a wider ecological context. And so I think having simulated environments where you can test things like co-scientists, for example, or an agentic system in a clinical setting would be really valuable to really understand, almost having a sandbox space where you could have benchmarks that are not really like today's benchmarks, which is very much leaderboard performance benchmarks, but actually more grounded in the biological truth of the outside world.
Yeah, it must be very challenging to create benchmarks when you've got a completely different data set, a completely different model. I can imagine there's a lot of hoops that need to be jumped through to get
Another
there.
way of going for it is to create competition. That's worked very well. People getting together, comparing for one shot. It's usually start like that. You compare your model performance for one competition, like basically on protein structure back in the days. On my side, I mean, one thing we are missing and we should improve is my point of view. So I'm the business development lead for Europe, Middle East, and Africa. And I'm obliged and I'm really faced every day with the comparison with my colleague and my managers and the entire NVIDIA operation in the US. What the energy is over there, the amount of basically investment going on, and the same thing happening basically in Asia, in China, and it's fantastic to see. It's very large, and they behave as a unit. And what we should improve in Europe is basically the ability to invest across borders, the ability to create companies in a very simplified manner, like in the US. We need to simplify this bureaucratic approach, allow investment to flow from, for example, the UK to Germany, and Germany can invest in Sweden, and Denmark can invest in France, and things like that. At the moment, it's hyper complex. Tax, regulation, laws are in different language, and they don't work with each other, and they are not compatible at all. So it's a true nightmare. And that's why we have, in fact, a trouble with that. We cannot attract investment from the outside, even within Europe. And it's like, it drives me mad. It's like thinking about Boston investors would just invest in Boston. You know, they would never look into Cambridge. God forbid, you know, it's too complex. You know, it's nuts. You know, it's just like California would never invest in a company based in Oregon. It's like, why? And we do the same in Europe.
Jonathan, what do you think we're missing?
I think one thing we could have talked about as well is regarding the teams that are beyond data and model and application and benchmark and investment, everything. And I think now we are, you know, not so long ago, if you go into the drug discovery spaces and you have to develop a drug, people there are just biologists. That's it. And if you come with a mathematician background or whatever, you do not even have the right to speak in the room. You have not. It's not where you should be. And now we come to a place where people with basically very limited biological background can still have a very interesting insight about biology. How do we deal with that? And those guys are extremely young as well. How do we deal with that? And I think now one of the challenges, something that we probably have as a challenge, is like, how do you build a team with people we still need the expertise in biology, in medical, in medical expertise as well, but we also need people that connect with biology from a mathematical point of view, and we need to do in both directions. And creating those teams with extremely interdisciplinary but native is quite a challenge, generation challenge, skill challenge, mindset challenge. And yeah, I think maybe the manager will have some challenges as well to build those teams in the future.
Wonderful, thank you. Well, I promised you that we would let the AI speak and let the AI join our panel discussion. So if we can put up the slide that has the response from the AI, we're going to see the first response here. So this was a question about asking the AI to predict what would be the impact of AI in the field of biology over the next 10 years. So I'm just gonna read it out. This was the first prediction. By 2035, biology will be an intelligent ecosystem where AI, labs, and living systems learn together. Scientists will code in cells as easily as in Python, designing therapies, materials, and ecosystems through self-improving models. Policy will lag, but shared norms of openness and safety will keep innovation both daring and humane. Any real brief comments on the AI's prediction? Yes, Jonathan.
Why do you invite us on the panel?
Okay, let us bring up the second prediction, please. So the second prediction, biology will become programmable like software. AI will predict cellular behavior with precision, enabling on-demand organism design. We'll see AI discovering entirely new biological principles we haven't imagined yet, not just optimizing known pathways. The biggest shift, biology teaching AI new computing paradigms through living neural networks and biocomputation. Any quick reactions? David?
I don't know about biocomputation and living organism as computer, maybe, yeah. I think we are going to composite computing sometime. I mean, we see quantum computers popping up and being hooked to, let's say, traditional accelerated computing and silicon computing. So yeah, why not? Yeah, I think quantum will have a role in the future.
Okay, let's bring up the third prediction from the AI. By 2035, my 2035 forecast, the magic wand tool we all want, a true digital twin of a cell, will exist, but our moonshot vision won't be technical. It will be the global light touch norms and shared culture we finally built to manage its dual use risks together. Wow, philosophical AI.
Well, I'm not sure I've understood everything out the end, but no, but that's very interesting. As I like to say, you can use those digital twins either for building drugs or building poisons. So basically, you have to choose your camp.
David, any thoughts?
I like the fact that it's mentioning dual use, because any technology which is getting extremely powerful is also basically dangerous in the wrong hands. But that's always been the case, anything.
I'm curious what the panel thinks in terms of prediction B about biology becoming programmable like software. I think amongst the iGEMers here, the iGEM community, the religion is that we're going to be able to program a cell just like we can program a computer. Amongst the hardcore biologists, there's still a lot of skepticism that that's even possible, that a complex system like biology can be programmed. And it takes me back a little bit to Christina Agapakis' essay that I mentioned about maybe it's not going to be programmed like a computer, but the AI is going to come up with new genetic designs that we don't necessarily understand, but still perform a particular function. So I'm curious about that paradigm of programmable biology. Maybe yeah, Sana first.
I think so. I think biology, to a large extent, in some sense of the world, is programmable already. And like CRISPR allowed base editing, prime editing has allowed us to program it in a way that we couldn't before. So I think every few years or decades, we might see a milestone moment that allows us to just program it more in a different way. So I think maybe not exactly like software, but I think maybe the software has more to learn from biology because we still don't understand biology. We understand software better rather than the other way around. But I think you're right in that it might unlock certain paradigms or creative spaces that we just hadn't thought about before. We have LLMs able to generate like green fluorescent protein alternatives, and we've seen that play out. We've got new bacteriophages being developed. We've got two antibiotic AMR-resistant sort of candidates in almost going into trials. So I think there is going to be an element of programming there. I guess for me, a lot of this sounds like really like pie in the sky to some degree. It feels very like politicians' way of saying, and then we'll all live happily ever after. So that's my only comment to that.
David?
I like the prediction. It shows a bright future for any computational biologist, AI engineers, medical device engineers, basically anybody will have a bright future in healthcare. It's going to be fascinating as a career. Second is that what it makes me think about is that it's a tale of caution. It's changing something in a living cell or a human body is definitive. So changing DNA, correcting errors, etc. I mean, we know from the past, it's basically it has a name. People try to improve traits in plants, in humans, and get rid of some things, but we need to be conscious of, what is our evolution? And potentially, those algorithms will need to take a long-term view of what would happen over a generation if we do that. Maybe that's, in fact, the next level to try to think about.
I love it. Let's give the AI a round of applause, but let's also give our three panelists a big round of applause as well. Thank you all for joining us today.